botany. And he also was in my same PhD lab. So a lot of what you're going to see today overlaps with things that I've studied in the past and love to geek out about. He's just really successful with them. You're going to notice this theme and speakers that I invite that many of them are on what's called the theoretical ecology tree of life. And this starts with Cy Levin. And I was very proud of myself for getting on this tree. And then Topher was with me at the bottom of this tree. And then for his postdoc, he leapt above me and went and jumped several layers above on this tree. So he's going to hear me reference this tree of life a lot, but Topher's a proud member of it. So anyways, Topher.
All right. Well, thank you, Ty, for that introduction. And I hope the talk kind of lives up to it. So I'm going to be talking about a few different aspects of my research in my lab. So from genomes to communities, which is my best attempt at sort of linking these two very disparate areas of research that you'll get a better idea of what I'm talking about in a second. And the idea of just generating these mechanistic insights. And that really comes from some of my thinking when Ty first invited me for this, he kind of mentioned that idea of wanting to bring in the idea of connecting theory with data and sort of connecting theory with big data. And so I was kind of thinking about how best to fit in this because just to put it right up front, I don't often consider myself first and foremost like a big data scientist. That's not how I tend to identify. I do end up using it some, but it's not sort of like first and foremost in my mind when I think about this. So what I tend to think about is the idea of connecting theory with data and how we can best do that. And so I'm going to be talking about two different vignettes in this talk. The first one is one avenue my lab uses to make this connection between theory and data. That's using microcosms and genomics to better understand eco-evolutionary dynamics, in this case in range expansions, but in a variety of sort of spatial contexts. Yeah.
Could you reshare your screen? It's actually not updating on Zoom. The slides, it's just on the first slide.
Interesting.
You just stop sharing and reshare. Probably.
Yeah, hopefully that would fix it.
Do you mind if I come up? I don't mean to.
No, please do. Yeah.
I think if we can see where's the mouse. It's actually pretty hard to see.
I know I was having trouble.
I was kind of my oh, here it is. Okay.
Yeah.
If we do this, can we drag? Where's your other monitor usually? Lower right, lower left? How do you?
Yeah. I think it's to the should be to the left.
Top. Anyways,
Still over.
I know it's because you don't have your screens like swapped, so it's like trying to figure out. Okay, you're going to be better at this because you work on your computer. I was trying to move your screen share over here so it'd be easier for you to navigate.
Yeah. Okay. Let's see. Is that?
Yeah, it's updating on Zoom now.
Okay.
Thank
cool.
you.
No worries. Technical issues averted, hopefully for now, for a slide or two. Okay. So first vignette, yeah, it's going to be talking about how we use microcosms to connect theory and data in terms of range expansions, particularly thinking about eco-evolutionary dynamics using some genomic analyses. And then just fully switching gears in the next section, we're going to be talking about some other work my lab does, developing sort of novel statistical techniques to estimate species pair interactions in diverse communities, particularly with annual plants and in that context, but it extends to other areas of community ecology as well. But first one, we'll just dive into this use of microcosms here. And the first thing I want to address is just why range expansions, kind of give a little context for why we care about this as a sort of motivating question. And one answer is that these are important historically because they're often the processes that have resulted in the current patterns of biogeography that we see around us today. So this is one example of this, where at the height of the last glacial maximum, spruce trees were sort of confined to these southern refugia, shown on the left side here, in those dark green colors. And then as that glacier receded, those spruce trees were able to expand their ranges northward, forming the sort of current distribution that we're familiar with today. But in a lot of ways, my sort of interest in range expansions goes beyond just thinking about this historic explanation for biogeographic patterns into thinking about more pressing conservation issues that we're facing today, like invasive species that are obviously one major area of range expansion that is quite concerning for a lot of conservation issues. So this is one example here showing cane toads expanding through Australia between 1940 and 1980. And since 1980, this expansion has obviously continued, and they're actually basically all across the northern stretch of Australia now. And this species is highly problematic for this ecosystem, causes a lot of devastating consequences for the local biodiversity. And so being able to understand these range expansions and be able to predict these range expansions can be really important and valuable for conserving those species. The other context that we've learned in recent years can be quite important in terms of range expansion is the idea of thinking about range shifts with climate change. So this is a graph from a 2011 paper showing observed latitudinal shifts among this particular graph showing butterflies, but you can see it across different taxa. And you can see on this graph that the distribution of shifts, there's kind of a range, it's somewhat variable, but in general, we're seeing shifts towards these positive values in terms of the shifts in these latitudinal ranges, which is positive in the sense that they're moving towards the poles, whether that's depending on the southern or northern hemisphere, they're just sort of moving away from the equator towards the poles in response to ongoing climate change. And this is, again, from a 2011 paper. These patterns have gotten more extreme since then. And so we've sort of realized in the last few decades that there's some really important aspects in these range expansions that we need to better understand to make these predictions. And in particular, there are some sort of underappreciated until recently evolutionary things going on in these range expansions that can really affect their dynamics. So to explain what I mean, thinking about these eco-evolutionary dynamics in these range expansions, I'm just going to show a bit of an illustration here, thinking about a sort of simplified range expansion in a single dimension. So moving through space on the x-axis, you'll see population abundance on the y-axis, and I'm labeling two different locations for this range expansion. First is the core, which will be on that left side of the graph, sort of stationary in space. And then the edge will be the shaded red region that you'll see sort of moving forward in time. And this graph is sort of illustrating two key points here. The first is that this edge location stays at relatively low abundances, low densities through time. And so it's characterized by these sorts of low population sizes. And each generation sort of represents this new founding event where you're getting this new edge population that extends beyond what's currently in the range via dispersal. So those two aspects are going to really inform these evolutionary dynamics that can occur in range expansions. And there's two key processes that I want to talk about. The first is this idea of spatial sorting. So this is an idea that was first talked about in this paper in 2011. And it's this idea that spatial range expansion can result in the evolution of increased dispersal. So how that occurs is if we consider a starting population, sort of this first generation of range expansion, if you will, shown with that graph previously, they might have an annual dispersal distance shown on the x-axis represented by this green distribution. But then as this expansion moves forward in time, the edge population that is formed now, sort of by definition, represents these individuals that have dispersed furthest from that starting population. And if dispersal is a heritable trait, these individuals that have dispersed high distances, they mate with other high dispersing individuals, they're likely to leave offspring with high dispersal abilities. And you can imagine how this process can sort of expand and continue through time to lead to this evolution of increased dispersal abilities at the edge of this expanding population. And so via time in these range expansions, you see, or you could see, evolution of increased dispersal abilities at the edge. The other key process that can occur in these range expansions is the process of gene surfing. And gene surfing operates via a founding effect. So just to kind of remind everyone what I mean when I say founding effect, if we think about it like a sort of starting population here, represented by these sort of cartoon genomes where these colors represent different alleles. If there's some sort of founding event that randomly samples a few individuals from this population, say the gray and the blue allele here, and then given time, a descendant population arises from that founding event, you can see that this descendant population now has a very different frequency of these alleles than the original population. And this can arise purely just due to random chance as sort of a sampling process here. And why this is so important in range expansions is because, as I said, through time, that edge population, essentially each generation represents a new founding event. And so if we start with this sort of initial population, and then the range expansion proceeds and sort of by chance, say this blue allele ends up at the edge population, it's likely to leave other descendants with the blue allele at the edge, and those individuals are more likely to be involved in future edge populations. And so we can get this circumstance where this blue allele surfs to high frequency in these edge populations, at least much higher frequency than seen in the core or in the founding populations. Now, this leads to a bit of a problem, though, when we're thinking about both of these processes occurring simultaneously, right? So now to illustrate this kind of dilemma that we face, I'm showing another graph with space along the x-axis, population size on the y-axis. Each point here represents an individual now, and these individuals for simplicity, are defined with essentially two loci, one that defines the shape of the point, one that defines the color of the point. And if we consider a range expansion proceeding through time, from this sort of pre-expansion period to this late expansion, we can see at the edge now, we have a population that's defined primarily by the blue color allele and by the triangle shape. Now, with a single range expansion, it's really hard to tell without like further analyses, further tests, which one of those traits, if either of them are associated, say, with increased dispersal, or if one or both of them just happen to surf to high frequencies by chance in that early range expansion. We can address this though if we're able to replicate the range expansion multiple times. So if we can sort of replay that same range expansion, we might see an outcome like this, where consistently we're seeing, say, the blue color at the edge, but now we have, say, the square shape or the diamond shape reaching that high frequency. And this allows us to kind of tease apart, just looking at these patterns, like, okay, maybe this blue color is associated with, say, dispersal or some other trait that's sort of selected for in the expansion process. And these shapes are sort of more due to just neutral fluctuations due to that gene surfing mechanism. And so being able to replicate this process is really, really important to tease apart gene surfing versus spatial sorting. So that's what led us to some of these microcosm experiments. And so when we set up these experiments, we had a few sort of driving questions. The first being just how important are these eco-evolutionary processes to range expansion overall? And this is because, these are, potentially interesting and like, it's kind of cool to know they're going on, but like, do they actually matter in the real world? Are they going to like actually affect the overall dynamics of these range expansions? Or are they just sort of a neat curiosity? And then furthermore, can we detect evidence specifically for spatial sorting versus gene surfing? Can we differentiate those two? And then finally, can we quantify the contributions of each process to their overall effects of evolution on range expansion? And better sort of incorporate them into some sort of predictive framework. So to answer these questions, we used Tribolium caspanium experimental systems. So these are the red flower beetles kept in these microcosms. Shown on the left here, you may have heard about these some from Ty, because as Ty mentioned, he's done a lot of work with this system as well. They're kept in these individual sort of acrylic patches filled with a flower medium that serves as a food source for these beetles. These patches have holes drilled in the side so they can be lined up into these linear landscapes. And so we can form these sort of replicated expansion processes on this very small scale that we can very tightly manipulate and control conditions with. And so this is work that I did as part of my PhD dissertation here at CU with Brett Melbourne, and then also in collaboration with Ruth Huffbauer at CSU, shown here. So they provide a lot of insight into sort of helping design these experiments, work through some of these analyses. But in these experiments, essentially what happens is these beetles are kept in these patches for most of their life, for most of a generation. And then for a sort of set period within that generation, we allow dispersal throughout these landscapes. So the beetles can kind of freely move throughout the landscape during this sort of dispersal phase of their generation. After a set amount of time, we cut off dispersal among these patches again, and then we're able to census these populations so that we can sort of get a precise count of how many beetles are where in the landscape.
Sorry, when you're saying landscape, you're talking about like this plastic box that you've created.
Yeah, there's a series of those plastic boxes. Yeah. You'll find, especially when I talk about these microcosms, I use the term landscape in a very not real sense, but it's a microcosm landscape, which works for my purposes. So apologies for any confusion.
And there's nothing, it's like, there's nothing in that box. It's like, other than, like, there's no, like topography or anything like that. It's just like a box.
Yeah,
okay.
No, this is a very simplified, just
Okay.
Each patch has a little bit of flower media to serve as a habitat, and then it's just a series of these patches. So it's incredibly simplified so as to allow us to get at like these, sort of direct questions of like, what is happening in an evolutionary context? Like, how are these expansions proceeding without any sort of confounding influences? And so to directly get at that question of how these evolutionary processes are being affected, we used two treatments. So the first that I'll talk about is called the shuffled treatment here. And essentially what we did is all of the beetles from a given landscape, we took them out, mixed them together, and then randomly redistributed them in these landscapes, but according to the numbers that we census them at. So in this way, we sort of broke any eco-evolutionary feedback present due to the spatial structure in that population, but we maintained the demographic structure of these range expansions. So we were able to directly sort of remove the influence of spatial evolution. And then in the other treatment, which I'll call the structured treatment, we simply just put the beetles back in the patches that they came from after that census process. And so they were able to maintain that spatial structure. Essentially their genotypes are sort of coupled to their location on the landscape in a way that the shuffle treatment sort of broke into that structure. I also want to mention, this is an incredibly labor-intensive process, maintaining all of these microcosms. So we had a lot of research technicians helping us with this process, including, I wanted to use this photo specifically because, so Adam Salon, who's on the left side of this photo, he was helping us with this experiment as an undergrad when he was here, but he's actually now getting his, I think his PhD in the eBio department. So just a shout out to Adam for all of his help, as well as all of our other lab techs. We ran this experiment for 8 generations. And these are some of the results we saw. So this is kind of just the raw data form, almost looking at distance spread on the y-axis, generation on the x-axis here, structured treatment on the left, shuffle treatment on the right. Each line represents one of our experimental landscapes. So we were able to replicate these about 30 times per treatment. And so looking at this, you can see there are some differences among these treatments, but obviously just looking at the raw data, it's a little hard to tell exactly what's going on. But when we look at mean distance spread through time, we can see there is a significant effect where these structured landscapes where they're allowed to evolve do spread a little bit faster on average. But interestingly, it's a relatively small effect. This only led to about 6% faster spread in these structured treatments. What's really interesting, I think, though, is that when we look at the variance in distance spread, these structured treatments have much, much greater variance compared to the shuffle treatments. We actually see about double the amount of variance after just 8 generations of spread. And so something about this evolutionary process is leading to this dramatic increase in the variability of spread rates, as well as a slight increase in the mean. So going back to our questions, we can say that, think about the overall importance of these evolutionary processes. We do see an increase in both the mean and the variance of this distance spread, but the largest impact is definitely on the variance. And this is, I think, particularly important and particularly interesting because this affects our ability to actually predict these range expansions. If these evolutionary processes lead to just a much greater variability in the outcome, this essentially means that we have a lot more associated uncertainty in these predictions. And particularly if we're making predictions from purely ecological models, it's uncertainty that we're kind of unaware of currently. So it's sort of meaning there's more uncertainty than we are currently accounting for or representing in our predictions for these range expansions. So I think that's very interesting. And I think it's important to kind of understand where exactly that's arising from. So we have this overall effect of spatial evolution. Now we kind of wanted to dig into, can we detect evidence for spatial sorting and gene surfing specifically? So to do this, we used a common garden experiment where after 8 generations of spread, we took 20 beetles from the core and the edge of our structured landscapes, and then sort of 20 random beetles from the shuffled landscapes where there's no real meaning behind the spatial location. And then we allowed them to reproduce in common conditions and use their offspring to test for evolution of some key traits. So in particular here, I'm going to show our results for dispersal evolution. So here we quantified dispersal as simply the probability that beetles would leave their natal patch after being raised there. And we did this at both high and low densities because the system is known to have some density dependence in dispersal. So at high densities, we didn't see any difference among these populations in their dispersal probability. Basically at high densities, all the beetles are trying to get into new patches to find unoccupied territory and to find some, find some alone time. But at low densities, these edge populations do actually show a higher probability of dispersing compared to these others. And so this was really interesting, especially because if you remember, these edge populations are characterized by being at low densities. So this is going to have some real sort of meaningful impact on the dynamics of these range expansions and likely accounts for that increase in the overall average distance spread that we saw. If even at these low densities, these edge populations are more likely to still be leaving and exploring new territories. So we do see evidence for spatial sorting in terms of increased dispersal probability at the edge. To get at evidence for gene surfing, though, we had to take a bit of a different tack, and this is where the genomic data comes in. So these are analyses that were done with a lot of help from Silas Titus and Nolan Kane here at CU. And essentially what we did is from these landscapes, we saved beetles from the first generation, from this generation 0 for later DNA extractions. And then at generation 8, at the end of the experiment, we took from the structured landscapes, those beetles from the core and the edge, saved them for DNA extractions, and then a random sample from the shuffled landscapes for further DNA extractions as well. So this kind of gave us this structure where you have this generation 0 or founding generation of beetles for both structured and shuffled landscapes, and then a core and edge sample from structured landscapes in the last generation, and just a random sample for comparison from the shuffled populations. So we extracted the DNA from all of these, we filtered for quality control, quantity of DNA, et cetera. This left us with 22 structured landscapes and 15 shuffled landscapes It actually left us with a little bit more than this, but we subsampled down to 96 populations. So these are 96 well plates. That was cheaper than doing just a few extra individual ones. And we did whole genome sequencing for this. So Tribolium castanea is a really nice organism for this study because it has a fully annotated reference genome available, and they actually have a relatively small genome compared to a lot of other organisms. But this many populations of whole genome sequence data does lead to a lot of data, right? So this is about 200 gigabytes of data, which I'm purely throwing up on here because, as I said, I'm a little self-conscious about being in big data seminar. So like, just trying to make a point that I do have access to big data. Maybe, I don't know what you guys think about 200 gigs. But the other point here is that this is a lot of data to go through and analyze, and I'm not going to have time to talk about all of the stuff we're doing and that has been done with this, but I'm going to go over a few key points with this in the context of some of our work. I also want to mention, obviously, this was a lot of work in terms of the extractions themselves, and this was another area where one of our lab techs, Tori Davies, did all these extractions for us. She was amazing. She did a fantastic job, so I definitely want to make sure she gets a shout out for this. But to look for a signature of gene surfing, what we did here is we looked at nucleotide diversity across the genome as sort of just a first pass here. And so specifically, if we think about these genomes and our linear chromosomes, we can split these into fixed windows and calculate nucleotide diversity in each one of those windows. And because of the structure of data, we have the founding populations, we have generation 8 populations, we can directly look for changes in nucleotide diversity specifically. And so we can quantify those changes over those 8 generations of range expansion. And this is important because any allele that's associated, say, with dispersal, that's sort of being selected for high dispersal at the edge, anything that's surfing to high frequencies, we would expect that to lead to a decrease in nucleotide diversity in that window containing that allele, because it's essentially increasing in frequency, it's excluding other alleles, potentially even reaching fixation. So we expect both of those processes to lead to decreases in nucleotide diversity, which again leads us to think about how can we differentiate things that are changing due to spatial sorting versus gene surfing. And so this is where we leverage that replicated nature of this experiment and of all our different populations here. So
can I actually ask you to clarify that language? So surfing versus, what was the two terms you just used?
So spatial sorting and gene surfing are these two.
Yeah, can you define those for us? Because I think a lot of us in this room, at least I don't know exactly what those mean.
Yeah, so spatial sorting was that process by which dispersal can evolve higher due to simply the just assortative mating of high dispersers at the edge of a range expansion. Gene surfing arises just from these repeated founding effects at the edge. So it's a purely neutral process, which is a key point there. So gene surfing leads to similar patterns where alleles can reach high frequencies, they can even reach fixation, but it's purely just due to this sampling effect. So it's entirely random, it's not due to sort of any fitness benefits that an allele has or any sort of action of selection, it's just purely random. Versus spatial sorting, it's specifically tied to dispersal ability. So we would expect that to sort of replicate across these populations. And so for that reason, when we're comparing across populations, if we're thinking about, say, a window like this, where we see consistent decreases in nucleotide diversity across all of our populations, we'd be much more confident in saying this is likely due to an allele associated, say, with dispersal or some other thing providing a benefit at the edge being acted on by selection, because we see these consistent changes across replicated populations. As opposed to windows like this, where we see sort of different changes in different populations and different replicates, we see it seems kind of random. These are likely changing due to the actions of neutral mechanisms, particularly gene surfing in this case, because we're talking about a range expansion. So by comparing across our replicates, we can sort of identify things that are likely being changed in individual replicates via gene surfing, because they're sort of different across replicates, versus things that are likely changing via spatial sorting because they're conserved and they're similar across all these replicates. And so to directly quantify that, we looked at the variability of these changes in nucleotide diversity across all the different chromosomes in the Tribolium genome. We compared that between core, edge, and shuffled populations. And so as a reminder on this graph, these larger values would indicate then a greater role for neutral mechanisms or for gene surfing in particular, because it's essentially indicating that there's a lot of differences among these replicate populations in these different windows. And so this is sort of what these results look like. So for the X chromosome, we see there is in fact sort of a larger value associated with these edge populations compared to the core and the shuffled populations, which is consistent with our predictions for how gene surfing should affect these changes. And in fact, we see that across the genome as a fairly strong pattern, that these edge populations show a much greater degree of variability in these changes in nucleotide diversity. So this seemed to us like pretty good evidence that gene surfing is causing these sorts of very random changes among populations and causing some important changes in terms of this genomic evolution. So in particular, we can say that gene surfing does seem to lead to this increased variability in genomic changes at the edge populations. And this is important because, again, this mirrors that increased variability we saw in the overall spread dynamics. This suggests that this gene surfing mechanism could be quite important in explaining why we see much greater rates of variability in distance spread in these populations. So last thing I want to touch on is then kind of really digging into that question of thinking about how does each process contribute to the overall effects of evolution and range expansions. And this is where we're getting into some of our ongoing work in the lab. So these are going to be sort of preliminary or just sort of, suggestions of what will be coming out of this. But to frame this a little bit, I want to go back to some of the results from the common garden experiment that we did to analyze evolution and traits. And in addition to dispersal, we also were tracking growth rate in these populations to look at changes in fitness. And a really interesting thing we saw was that these edge populations have much lower fitness after range expansion compared to core and shuffle populations. And this was kind of confusing for us for a while because we weren't sure what exactly was going on or why there should be this lower fitness at the edge. Until we did some more digging, we found these theoretical papers looking at the effects of gene surfing that suggested that because it's this entirely neutral mechanism, it can lead to sampling of deleterious alleles that get associated with edge populations. And because it's this repeated sampling process, selection is very much reduced at the edge. And so these deleterious alleles can still just kind of surf to those higher frequencies or even reach fixation because of those repeated bounding events. And because it's just repeated sampling, this low population sizes, all of that combines to sort of reduce the efficacy of selection against those alleles. And so in theoretical models, at least, it suggests that there can be this accumulation of deleterious alleles at the edge. And I should note, we also looked into whether this was due to sort of trait correlation between increased dispersal and reduced fitness. And there's no good evidence to suggest that these two are linked, actually. So this seems to be an uncorrelated event, so not like a trait trade-off or something like that. So we kind of were left with like thinking that maybe this is due to that accumulation of deleterious alleles. But we wanted to try and find some better evidence than just sort of a matchup with this sort of theoretical prediction. So this is a project that my current PhD student, Spencer Holtz, is working on right now, trying to identify a signature of potential buildup of deleterious alleles in these populations. And so for this project, he's using this technique called genomic evolutionary rate profile. And so what this means is he's building a phylogeny of these beetles related to Tribulum casmium, and then using that phylogeny, kind of assessing rates of neutral evolution at various locations in the genome and identifying areas where there's more rejected substitutions than you would expect by purely neutral mechanisms. And essentially what this allows is to identify areas that are putatively under some type of purifying selection for a specific allele value that seems to be conserved in this clade here. And this is an ongoing project. He has a little, he had a background in some genomic stuff, but this is, a lot of new things that he's learning right now. So it's, and work in progress. We only have some preliminary results right now, but it does look like when we look at some of these sites that are identified as being putatively conserved, that these edge populations actually do have higher degrees of these derived alleles at these conserved sites, which would indicate that there's potential for this buildup of these mildly deleterious alleles reducing fitness at the edge. So we think that we're going to find some pretty good evidence for gene surfing leading to that buildup of deleterious alleles, potentially reducing fitness at the edge. So that's one outcome that we're sort of hoping to get more on soon. Yes.
Can I ask you to actually back up a little bit on that? So when you say he's building out a phylogeny, what does that mean? And like, how does he do that with his data?
So essentially, the phylogeny is built on available genomes through GenBank. And so we look for any sequence genomes that are available from related species. And then you can use various genomic alignment softwares to create a phylogeny of sort of like suggested relatedness. We validate that via other published phylogenies that are based on just sort of subsections of the genome or traits even and things like that. But having that in hand, then we have like all of those genomes aligned together, and then we can sort of look across them and compare to find those sections of the genome that are seem to be conserved to varying degrees of strength. Does that kind of answer that question? Okay. So he's working on that right now, but it does seem like there's some good evidence that we see some of those deleterious alleles buildup in edge populations that could be responsible for that decrease in fitness at the edge. The other key thing we're working on right now is trying to really quantify how spatial sorting and how gene surfing and then their interaction contribute to that variance we saw in range expansion. So if you remember, I sort of suggested that increased variance and changes in nucleotide diversity kind of mirrored the increased variance we saw in overall spread rates. But we don't have a direct link between those yet. And both spatial sorting and gene surfing have the potential to sort of increase variance in range expansion simply because there are additional processes at work. So there could be variability in how efficient spatial sorting is, or gene surfing leading like different alleles to fix at the edge and things like that. So we really want to try to nail down that, quantify how much strongly each one of them contributes to that. And so this is work my postdoc, Brianna Powers, is working on right now, using this SLIM software, which is a forward genetic simulation software. Essentially, it's an agent-based model, but tracking both genomes as well as individual location, traits, things like that. This software was developed by Haller and Messer in 2016. It's been updated several times since then. And what she's doing, she's building different models that incorporate either spatial sorting, gene surfing, both or neither, and then fitting those to our data using approximate Bayesian computation. And then using that to try and figure out which one of these sort of best explains the data, and then directly kind of being able to turn different processes on and off to better understand when we see that increase in variability and when we don't. And so again, this is a bit of an work in progress. So like she's kind of creating these models that just create sort of similar dynamics to what we saw in the experiment of spread through time, and then trying to understand how these processes influence variability in that model. So that's an ongoing thing. I don't really have results to show from this yet, but she will be presenting this at ESA this summer. So anyone who's going, I encourage you to find her poster, and she'll hopefully have some good results by then. Yeah.
And just clarity, your data here is the same data from those plastic landscape things, or where's the data for this?
Yeah, it's all the microcosm data. So the range expansions, the genomic data. This software, as I said, it tracks both genomes and those sort of individual locations and traits and things like that. So we're able to fit both sort of population abundance patterns, spatial spread patterns, and then changes in genomic patterns as well all through this software. So how many do you have? Okay. So this is kind of where some of our current analyses are at. Looking into this data, I'm happy to talk more about this with people who are interested. But again, I kind of framed this as we have like these two different vignettes to talk about. So I'm going to now switch gears entirely to talk about community ecology and our work there. But I'm happy to talk about any questions from the first section or the last section, whatever kind of comes up. But now we're going to move into talking about developing these novel statistical analyses to quantify species interactions in diverse systems. And to kind of motivate this, I want to give a bit of a background on how modeling population dynamics works in community ecology from a theoretical perspective. So this is the Beverton-Holt model. It's very commonly used to model population dynamics in a community ecology context, at least specifically for plant community ecology. I realize microbial community ecology is kind of a different ballgame that I'm not as familiar with. So apologies to any microbial community ecologists out there. This is going to focus on plants. But in this equation, essentially we model fecundity in a given environmental condition, which is the E subscript for a specific species, which is the I subscript here, as a function of a species' intrinsic growth rate in that environmental condition, and then scaled by the strength of intraspecific competition, so how strongly individuals of the same species kind of suppress growth of other individuals of that species, and then interspecific competition for all other species in that given community, how strongly they suppress growth rate of the focal species. And I bring this up because I just want to point out, you can see that this is going to start incorporating a lot of these different pairwise species interactions, these alpha terms. So essentially you need an alpha term for each interacting species in the community, as well as that intraspecific alpha term. And this can be kind of problematic with a lot of the data sets that we have in community ecology and lead to this kind of disconnect between the theory that we're developing and the data we have available, because estimating these species interactions can be quite difficult. And to illustrate that, I'm going to show how this works for just a relatively simple low diversity community with only 6 species. And so for this community, we need to estimate these intraspecific interaction coefficients, so how this species, say, affects itself in terms of fecundity and population growth, how the species is affected by all these other species in the community, and how they suppress the growth rate. And then we need to do that for all of these species, right? So just for the 6 species community, we're left with needing to estimate these 36 interaction coefficients. And that's if we're assuming these interaction coefficients are constant across space. But often we have these studies that are across environmental gradients, say, and we might expect the strength of these interactions to vary with underlying environmental conditions. And so in a given environmental gradient, we might expect, say, the strength of competition to, say, increase as we move along that gradient. But we don't necessarily expect that that should be the same for all of these different species pairs, right? We might have different species that are sort of less affected or more affected by this environmental gradient. And so even with a relatively simple model of interaction strength versus the environment, say like a linear model where we're estimating an intercept and a slope, now we need to estimate 72 coefficients to fully capture the dynamics of this community, right? And particularly for a lot of these plant ecology data sets, we simply just don't have that much data available often. So it's really, really difficult to get models to converge, even if we're not thinking about underlying environmental gradients, because it's simply just very, very difficult to get all these estimates to come out with these data sets when there's just not as much data available as we would like. And so this is a challenge for a lot of community ecologists trying to apply this theory in realistic settings. And so I want to give a shout out to this working group that sort of provided a lot of the motivation for this particular study that I'll be talking about. So this is the Estimation Working Group funded by the German Center for Integrative Biodiversity Research. And this Estimation Working Group was basically started with the goal of being able to apply theoretical community ecology concepts to sort of on the ground restoration settings. And how can we directly link that and better use theory to inform restoration practices? And specifically, I want to give a shout out to the three co-organizers of this group, Lauren Shoemaker, Lauren Hallett, and Akasha, who actually, as it happens, are all also CU Boulder alumni. So shout out to CU there. And then they started this working group pre-COVID. They had their first meeting. Obviously, things kind of took a pause for a while there. But one of the issues they ran into was like, how do you use these theoretical models that require all these pairwise interactions for restoration data? And that was when myself and then a postdoc that Lauren Shoemaker and I were co-advising, Chai Warner, kind of came onto this project to help develop some of these new techniques. But to illustrate the problem here that they were facing, I want to go over some of the existing approaches that get used in scenarios like this. And so the first one is to use a priori assumptions about this community to lump species together to kind of artificially simplify things. And these can look like a lot of things. One common thing is to lump things as sort of native versus exotic species. You also see this used as like grasses versus forbs or things like that. But you essentially lump species into these different categories, and then you make the assumption that, say, all native species affect other native species in the same way. So you kind of lump those interaction coefficients into a single value. And so from this set of potential 36 interaction coefficients, you'd be left with sort of estimating these 4 possibilities. But you can imagine why some people don't really like this approach, because this assumption is kind of a strong assumption to make, right? That like all these species are going to interact the same. So a lot of people don't really like that. It just gets often used as a matter of convenience. Another approach that people tend to sometimes use is to ignore variation among heterospecifics. And so what I mean by that is different species interactions, those interspecific interactions, kind of ignoring that variation to focus on these intraspecific terms. And this is a somewhat popular approach, primarily because these intraspecific terms are particularly important from a theoretical perspective, because they determine sort of stable coexistence often in these communities. And that's because when a species limits itself more strongly than it limits competitors, that's a sort of prerequisite for a lot of stable coexistence ideas. So essentially, if a species is going to self-limit more than it limits other things, it's not going to just fully take over and form this sort of monoculture. And so being able to estimate these intraspecific terms can be quite important from a theoretical standpoint. But then this approach sort of lumps all these other terms as sort of a single value for just like how any random heterospecific will affect conspecifics. And this, again, you can imagine is not as there's some problems people have with it for the same kind of reasons of just there's a lot of variation here. And you can assume that many of these instances wouldn't quite be the same. Sorry, is there a question?
Yes. So in the first, I'm looking at this like matrices, right? Mm-hmm. So in the first matrix, you have one species affecting the other species. Mm-hmm. And it's color coded to be different from the other species affecting, like it's not going bidirectional, like it's color coded differently. Mm-hmm. Is that intentional?
Yes, and that's essentially the idea that for these interactions, the effect of, say, species 1 on species 2 is not guaranteed to be the same as the effect of species 2 on species 1.
Okay.
So we're sort of allowing those two to vary. And so we're not just dealing with, say, a triangular matrix or something, because we sort of assume that just because one species is maybe strongly inhibited by another, that it would have that same inhibitory effect on the other. That obviously can happen, but it doesn't happen in a lot of cases as well. So we're kind of allowing for that variation. Does that answer that question? Okay. Okay, so this is another approach that again kind of lumps a lot of potential variability into just a single parameter. And so people aren't always necessarily very happy with that approach. The final kind of existing approach is to artificially simplify the community. This is quite popular with sort of theoreticians who are just trying to get some empirical data to test some of these models and these theories. And essentially what they do is acknowledge this variability exists within this community, that there are all these different parameters that can and likely should be treated as distinct parameters. But then they sort of ignore the vast amount of diversity in a system to sort of artificially just think about, say, a two-species system. This is often done via like weeding or exposure experiments, or even greenhouse experiments and things like that. But this obviously was not an attractive approach for this working group that's specifically trying to apply theory in a restoration setting, right? You can't artificially ignore all the diversity if you're trying to restore an ecosystem. And so this led to this kind of conundrum of like, how do we best apply these models to these diverse systems while recognizing that diversity? And that led to our approach, which was to adapt a Bayesian sparse modeling technique. And essentially what this means is that we adaptively identify parameters for inclusion in these models as part of the model fitting process. Now, I want to be sure I'm clear on this. This is something we are borrowing from existing techniques in computer vision, genomics, a lot of other fields. This is not a technique we sort of developed by any means. This is a technique that we sort of recognize the potential for and sort of adapted for these community dynamic models. But there's a lot of existing work kind of developing a variety of these different techniques for sparse modeling in general. But essentially how it worked is that we still kept all these intraspecific terms in the models to make sure we always estimate those, because as a kind of acknowledgement of, as I was saying, these are quite important from a theoretical context to be able to assess community stability and things like that. And then we allow these terms to be sort of generic and let the model decide which of these interspecific competition coefficients needs to be estimated as sort of distinct from this generic background term. Because a lot of these probably can be lumped together as sort of a species, is affected by species 1, 2, or 3, essentially in the same way, because it's essentially just like, oh, there's something else here and that's affecting it negatively. But there are likely certain species pairs that are distinctly sort of more or less competitive because they're maybe like competing strongly for the exact same resource or the same type of niche or things like that. And so during the model fitting process, the model kind of identifies which of these parameters to include as distinct from that background process. Now, I'm not going into the details of the sort of like the math behind this model and the stats. I'm happy to talk about that later with people who are curious. We're also pretty excited. This paper just came out like about a month ago in Ecology Letters describing this technique and how we used it for community ecology. So you can also check that out if you're curious. But I just want to go over a little bit of what we did with this model. The first thing was just to validate it with some simulated data, make sure it's sort of acting the way we hope that it would act with known parameters. And so that's what I'm showing here, where on the x-axis here, there's different size data sets that we used as input. And then on y-axis, we have parameter deviations for lambda, the intrinsic growth rate, for intraspecific competition terms, for generic interspecific competition terms, and then for the species-specific terms. And for each one of those, we allowed them to vary along a simulated environmental gradient. So we have a slope and intercept for both. And so what you can see here is that with 10 data points as a sort of input data set size, the model is estimating some terms fairly well, in particular the intrinsic growth rates. The others, it's doing okay, but there's obviously a lot of deviation because it's a relatively small data set. And then as we increase the sample size, we see increased accuracy in our parameter estimates, which is about what we would expect, right? So nothing necessarily earth shattering here, just more that like this is behaving about how we would expect. Some interesting dynamics that did come out of this, I think, are that one thing we see is that with increasing sample size, the model is able to pull out more of these non-generic terms, so sort of species-specific value for an intercept or a slope in these interspecific competition terms. And so you can see with only 10 data points, on average, the model is almost always just leaving everything as sort of the generic term and doing its best with that. And then as we increase the size of the data set, the model is able to pull out more of these things as sort of non-generic species-specific interactions. And in particular, I think that's kind of interesting paired with this last plot here, which is showing root mean squared error for the model making predictions of a species growth rate with out of sample data. And essentially what we're showing is that, again, as expected, as you increase sample size, you get lower root mean squared error. But what I thought was really interesting was that this drop off is actually less dramatic than I was expecting. Like the model does pretty well with these predictions, even with just 10 data points. And part of that is that it's sort of it's able to adaptively balance, depending on how much data it has available, whether it's worth it to try and estimate and include these species specific terms, or whether to just keep it as that generic term and sort of do the best it can with those. So if those low data set, sizes, it sort of doesn't bother to try and estimate those terms that would probably have a lot of error associated with them because there's not enough data to really estimate them effectively. And it just uses the generic term and it's able to do a decent job. And then as you get more data, it is increasing its predictive accuracy, but sort of the biggest gain that you get with that data is that you're able to pull out those species specific terms and you get a better sense of sort of what some of the underlying community dynamics might be in terms of identifying specific species pairs that are maybe more or less competitive. So we also then wanted to apply this to some existing data that we had available. So this is data from York gumdrum woodlands in Southwest Australia. And so these are woodlands with these York gumdrum trees and then a sort of understory of annual grasses and forbs primarily. And this is a nice system to test out this model with because there's already 2 environmental gradients that have gotten a lot of study in this system and have been predicted to have an important effect on that understory community. One of those gradients is shade caused by canopy cover from those trees. And then the other gradient is soil phosphorus. And that's because a lot of these areas are nearby these agricultural areas that have a lot of runoff from fertilizer and things like that. And so there's been a lot of study on these environmental gradients. And for this project in particular, this is using data that Kath Bowler collected as part of her PhD dissertation with Marty Mayfield. And then Lauren Shoemaker and I helped with the analyses. Isaac helps with a lot of the data collection, sort of background knowledge of the system. But so in particular, we collected data from these 2 sites, Western Perenjori Reserve and Bendering Nature Preserve in Southwest Australia. And you can see both these preserves are in slightly different rainfall regimes as well. So you got some variability there in that sense. And this community is about 44 species, depending on where you're sampling. As I said, it's annual is about a mix of grasses and forbs. And the study design used a system like this, where within a plot for a given focal species, Kath sort of randomly selected focal individuals, put down these interaction rings around them. And half of them that were randomly assigned to a treatment had to be thinned out to like remove all of the competitors, both the conspecific and the heterospecific competitors. And that was to get estimates of that intrinsic growth rate or growth and fecundity of the species without any competition. And then half were sort of left as is to get estimates for those interaction coefficients. And so this is a really nice data set to use for this also, because as I said, there's about 44 species here. So it's, you know, not even like as diverse as you might get from like some very diverse terrestrial systems, but it's more diverse than we could possibly estimate if we just tried to like directly estimate each one of these pairwise 44 by 44 interaction coefficients. Like there's no way we could do that with the data. And I know that because we initially tried to, and it failed miserably. So we wanted to use our sparse modeling approach here. And so I'm going to be showing results specifically for one species in this system, just to keep things a little simple, which is the Waitsia acuminata, which is an annual native forb in this system. And we allowed these interaction coefficients to vary across environmental gradients. We used a log linear transformation, and that's purely because for the Beverton-Holt model, those interaction coefficients have to remain positive. So we had to use that transformation to kind of keep them in the positive range of values. And then again, as I mentioned, Perenjori and Bendering are in these 2 different rainfall regimes. So we also allowed these to vary between those different reserves to allow for sort of potential differences, whether it's due to rainfall, whether it's due to just local adaptation or things like that in those different reserves as well. So for just this species, we had, you know, potentially for each pairwise interaction coefficient, we had these 4 values that we were potentially going to estimate from the data. So to show you some of these results, first, for intrinsic growth rate across the phosphorus gradient, you can see that in general, with increasing levels of phosphorus, we see these lower values of the intrinsic growth rate. But interestingly, you do see a pretty big distinction between these 2 reserves. This is the same species, but just in these 2 different reserves. And in Perenjori, we see this much more dramatic decline in intrinsic growth rate in response to phosphorus compared to Bendering. Now, whether that's just due to, say, the differences in rainfall regimes there, whether that's due to some sort of local adaptation among those 2 reserves, we don't necessarily know yet. But it was kind of interesting to see that reserve effect being so dramatic. The other thing we can see when we estimate our interaction coefficients is, first, I'm just showing across this gradient of phosphorus intraspecific competition and then our generic interspecific competition. And we can see that we get increased intraspecific competition with increased phosphorus, which sort of aligns with what's been observed already in this system, which is that if this increased phosphorus leads to sort of poor outcomes for a lot of these data species. And we also see that this intraspecific term is higher essentially across the gradients than the interspecific competition. And again, that's important from a sort of community context because that implies Weightia is able to stably coexist and not exclude everything else in the system, because it's going to be limiting itself more than other things are limiting it. But we do see that break down a little bit at that higher end of the phosphorus gradient, which is kind of interesting. And then we can add in our non-generic competitors to the model estimate. So in this case, we see two that get pulled out as non-generic. One, H-glutenism in Parangori is sort of has a much stronger competitive effect than a generic competitor. And then another, S-notus and bendering actually has a slightly weaker effect than a generic competitor. So this kind of allows us to get some more insight into how these community dynamics unfold in the system. You can do the same thing with canopy cover. And sorry, I realize I'm running a little late here, but you can see a similar result with canopy cover, increase in canopy cover leading to slightly lower intrinsic growth rates, relatively minor effects. And then we can also pull out these interaction coefficients, as well as some of those species-specific terms. Key thing I want to mention is using this method, we can then apply some of these theoretical contexts from community ecology to better understand the system. In particular, we can use these results to extend modern coexistence theory to diverse communities. So this theory is sort of based on understanding low density growth rates of species in a community to identify whether they can coexist in that community. So a positive low density growth rate implies that if a species gets knocked down to low abundance, it's able to recover, and so it's likely able to persist in that community. Negative growth rates mean that it's likely to be excluded. With our Bayesian framework, we can also look at this in terms of posterior distribution, so we can get a better sense of the uncertainty here. So we can say on average, these two species have positive growth rates and would be predicted to coexist. We can quantify sort of the extent to which there's this probability that one might be competitively excluded by being on the left side of this zero line here. I'm sorry, I'm going a little bit fast through this, so let me know if you have questions afterwards. So showing these results for Weightia, again, things on the left here would be probability of exclusion. Things on the right means probability of persistence or coexistence. And I'm going to go through a few different scenarios sort of relevant to restoration or conservation managers in this system. First is our overall distribution, which we can see indicates that Weightia can coexist in this system. It's primarily in the positive range here. There's some amount of this distribution in the negative range. There's maybe a very low probability of exclusion, or maybe some populations that are sort of only maintained to be a dispersal in this system. But now we can sort of use our model results to really get at direct manipulations of different parameters of the system. So for example, we can look at if we changed canopy cover or shade and show that kind of similar to the results I showed before, where there's a very weak effect along that gradient in terms of intrinsic growth rate. We don't see much of a change in terms of low density growth rate when we change shade. In contrast, when we lower soil phosphorus, we can see this big payoff in terms of Weightia being able to coexist much better here. And then we can also cross this with manipulating the competitive environment using those interspecific terms that we estimate. In this case, what we see is when we manipulate the competitive environment, we do get a benefit to Weightia, but it's actually less strong than we get with just phosphorus. And when we do both, there's not that much of an increase beyond what we get for phosphorus. So in this case, if we were to give recommendations for, say, a manager in the system, it would likely be that they should focus on soil phosphorus and not worry too much about some of these like sort of manual weeding treatments or spraying herbicides or things like that, because the biggest payoff is going to be in just reducing that phosphorus runoff. So essentially by leveraging these sparse modeling techniques, we can extend these traditional population dynamic models to diverse communities. We can then extend these theoretical concepts to diverse realistic systems, which I think is a quite exciting aspect of this. And importantly, we can then derive theory-driven management recommendations for the systems, which is a big sort of desired outcome from that restoration work group. Just to kind of wrap up both vignettes a little bit, some closing thoughts for my first big data seminar here. One, just kind of tongue in cheek a little bit, big data can come from these small places, these small microcosms. It's the biggest data set I've ever worked with, at least. But potentially more importantly, I think the second outcome kind of shows an interesting result here, which is that these big data techniques like sparse modeling and stuff shouldn't necessarily be restricted to just working with these big data sets. They can have really important results from working with sort of more traditional, small data sets or things like that. You can get these really interesting outcomes by using that. So I'm just going to acknowledge a lot of these people during my talks. I'm going to throw up, so I did in the working group, a bunch of my CU colleagues and lab techs helping with funding for that beetle experiment. University of Wyoming providing funding for my current student and postdoc, as well as what we worked on Kat's thesis work. And then I want to give a shout out to Modelscapes real quick. This is an NSF funded project. It's a collaboration among the University of Wyoming, University of Nevada, and University of Montana. And it's all going towards postdoc funding, specifically for developing statistical models, applying new modeling techniques to a variety of biological problems, ranging from aquatic ecosystem ecology, community ecology, genomic and evolutionary research. So if anyone is in need of a postdoc or will be in need of a postdoc within the next few years, we have a lot of postdoc lines to fill with this project. So just giving a shout out to that. And with that, hopefully I didn't go too fast through those last slides, but happy to answer any questions.
An online chat, you're welcome to put questions in the chat and then I'll read them.
I'll jump. I mean, I've been jumping in constantly. So we actually just got a project funded by NASA to look at eDNA and remote sensing data in South Africa, with the idea of like, how does biodiversity organize across scales? But so we are we're working with a microbiologist, so not an expert in this at all. But I guess I'm just wondering, like, if you could see some pathways to like how some of these sort of more traditional experimental setups might transition to a more sort of like advantageous kind of experiment of like eDNA across the landscape. I mean, one of the advantages to eDNA is that it's got this huge citizen science movement behind it. And so I'm just wondering if you see any pathways to like transition from laboratory to field.
Yeah, no, that's a really good question. And I think there are. I mean, you brought up the idea of the citizen science aspect of eDNA, and that's one thing working with range expansions in particular can be kind of nice because there's a lot of, not necessarily for a good reason, but a lot of charismatic invasive species out there that people tend to really care about. And so there are some existing citizen science projects on like sort of collecting data on just spread rates and things like that. And I think there's some that I haven't worked on this yet, but we're just starting a collaboration with some folks working on the expansion of tamarisk, which is kind of a nice fit for matching certain predictions from our microcosm system, which is obviously very simplified, using this linear expansion framework, but then tamarisk kind of like expands along these waterways. So in many ways, it's similar to sort of this one-dimensional linear expansion. And so we're hoping to kind of be able to find some nice matches between that and some of the predictions from these models. So I think there are some nice potential avenues there, but I'm like just dipping my toes in the water that way.
We have a question from online, Mike.
Hey, Tover, can you hear me?
Yes.
Great. Thanks for your talk. That's super interesting work. I've got a question about the first half as a fellow lover of the Tribolian system. I'm wondering about, I guess this like cannibalism rate has always been really intriguing to me as something that could potentially evolve or be more prominent in a like novel, hard or crowded kind of system. And I'm wondering if you've sort of thought about that component or how that might play into the, this sort of balance between the gene surfing or the spatial sorting kind of at the edge and the range expansion.
Yeah, that's a really good question. We've looked into that in some other experiments. So to give some context, the cannibalism rate in this system essentially is the mechanism by which density dependence operates in population growth in Tribolium. So essentially these beetles are crawling around in these patches, laying eggs, and then finding other beetles' eggs and eating them as a sort of protein source, essentially. And so this cannibalism rate sort of is really what regulates the overall population sizes in these patches. And there's been some other experiments that I didn't talk about today where we did fit explicit population models to look at, say, I think it was with founding effects. There's actually, you might know this experiment pretty well, Mike, because it's from Ruth's lab initially, but it was looking at founding population size versus founding population genetic diversity. And then we fit some models to that to look at how those factors affected both intrinsic growth rate as well as that sort of intraspecific density dependence. And we did see some indication that I believe it was low population sizes. Oh man, it's been so long. I think they increased the cannibalism rate actually. So that would indicate that potentially with this gene surfing, that could be another, or with range expansion in particular, where there's those low edge population sizes and those founding effects, we might actually see that change. But we haven't actually fit those models to this data. It's a really good idea to kind of look into that.
Interesting. Cool. Thanks.
Okay.
Yeah, thank you.
I have a question, then we have one more online. So you were talking really early on in the talk about the uncertainty and how just the variability was much higher than expected specifically with like the edge populations. And so I'm wondering, do you know, has that been adapted into current species model, like range expansion models or anything like that? Because I know we work a lot with those. And so it's definitely making me question. A lot of managers also want them because they're like, well, we don't know where they're going to be. We want to know where they're going to be. But if the models can't tell you, I mean, I just want to hear your thoughts about that.
Yeah, no, that's a really good question. And that's honestly, it's a hard question because I would love to be able to like fully kind of incorporate all of that too. And that's very much an active area of research at the moment, is trying to get that stuff figured out. One of the challenges is that, we still don't know exactly, like, is that increased variability due to that evolutionary dispersal? Is it due to gene surfing and things like that? And depending where exactly that's coming from, to fully incorporate it, like it might require a lot of additional information than what a lot of, say, species distribution models currently need. Because for example, if it's largely driven by gene surfing, then likely that means we would need to know information about like what's the existing genetic variability in the population, like how, like, what's the existing sort of genetic load in terms of like existing deleterious alleles that are present and things like that. Which is obviously not easy data to collect necessarily for a lot of these things that we want to be able to use species distribution models for. So it's an ongoing area of work, and it's not, unfortunately, I think, an easy answer at the moment, but we're trying to get there.
Yeah. So the question is from Isabel. She says, great talk and work. Gotta read it. How does your modeling of species interaction coefficients, both inter and intraspecific interactions, differ from joint species distribution modeling, say with the R package, HMSC, if you know? Let's see, maybe there are some key underlying community ecology theory differences in assumptions, say in how one models intrinsic growth rates.
Yeah, so that's a really good question. And I don't know that R package specifically, so I'm going to have to answer somewhat generically. But based on what I do know of a lot of species sort of co-occurrence modeling, it's a little bit more phenomenological, kind of looking at like how often things co-occur together versus not. And these sort of population dynamic models, because they're able to incorporate both the effect of intrinsic growth rate as well as these interaction coefficients, are able to tease apart in a little bit more of a mechanistic way. Like, do they co-occur rarely because one outcompetes the other directly, or do they just have sort of different intrinsic growth rates in those environments, so essentially different niche spaces? And so you can get a little bit more of that mechanistic detail if I'm understanding the species co-occurrence stuff that is being asked about. So feel free to correct me on that if I'm not.
We do have a recorded seminar on that package, if anybody is interested. That was Adam did that a few weeks ago, or months ago, really, at this point. Any more questions? I think we're good then.
Cool. Well, thank you, everyone. This is fun.
I just want to remind everybody that there is